Welcome back. Let's talk about glycolysis. Glycolysis simply means that during this process, we're lysing or breaking down glyco or glucose molecule. While we're going through each step, I want you to pay attention to a couple of things, including substrates and products, which I will color code with a cream color. I will show both products and substrate with this color because it's pretty challenging to color code substrates and products differently, since product of one step would be substrate for the following step. Thus, I will show them both with this cream color. Next thing to pay attention to is input and output, which for the purpose of this video, I will define them as electron carriers NAD+ and NADH, oxidized and reduced form, as well as ATP and ADP. Lastly, I will color code the important enzymes with a yellow color. Before jumping into details, let's understand the overall reaction, which I will write here on the right. During glycolysis, I have one glucose molecule. I will invest 2 ATPs and 2 NAD+, the reduced form, and what we get ultimately is 2 pyruvate molecules, 2 NADH, which is the oxidized form, 4 ATP molecules, and to be thorough, 2 H2O molecules. And now, it's time to dive into details. Okay, I have my glucose molecule with the formula C6H12O6. And what I'm going to do, or during the first step of glycolysis, I am turning 6-carbon glucose molecule into glucose 6-phosphate. By adding one phosphate group to my substrate with the help of enzyme hexokinase, and investment of 1 ATP molecule. Next, I will turn my glucose 6-phosphate, which still has 6 carbons in it because I haven't touched the number of carbons yet, and turn it into fructose 6-phosphate. Basically, we're isomerizing glucose to fructose, and the enzyme which is involved in this process is phosphoglucoisomerase. And I'm not writing it down because it goes into more details that you need to know for the purpose of DAT. But it is important for you to understand that we're not touching the number of carbons. We are just isomerizing it. Next, I will turn my fructose 6-phosphate into fructose 1,6-bisphosphate. Again, the number of carbons is untouched. What I have done is just adding another phosphate group to the other end of my molecule with the help of enzyme phosphofructokinase and investment of 1 ATP molecule. This step with this enzyme is the key in regulation of glycolysis. Once it goes through this step, it commits the cell to undergo glycolysis all the way down to the pyruvate. So I'm showing it with a one-way arrow to emphasize how important this step is. Next, I have my fructose 1,6-bisphosphate, which is very unstable due to attachment of 2 phosphate groups at each end. Aldolase enzyme helps this molecule to reach more stable energy level by splitting it into 2 molecules of pGal or G3P, which stands for glyceraldehyde 3-phosphate, and DHAP or dihydroxyacetone phosphate, both of which have 3 carbons in them. DHAP and pGal can interconvert to one another, but almost 99% of the time, pGal gets removed ultimately in the form of pyruvate, and according to Le Chatelier's principle, it causes the reaction to turn left as drawn toward pGal. In fact, This reaction never reaches equilibrium. Alright, then I have my pGal, which goes through 5 steps, each having their own intermediates and enzymes involved. And I'm not writing them down because they're not necessary for the purpose of the DAT. Ultimately, they will give me a pyruvate molecule. During these 5 steps, we are generating 2 ATPs and also investing 1 NAD plus to get 1 NADH molecule. Again, during these 5 steps, I am not touching the number of carbons, so pyruvate still has 3 carbons in it. One very important part of glycolysis is that you need to notice that per each round of glycolysis, we will get 1 pGal and 1 DHAP. But, as we discussed earlier, DHAP will turn into pGal. So we end up getting 2 pGals that ultimately gives us 2 pyruvates. And all the inputs and outputs will double during these 5 steps. That's why I have the number 2 NADH and 4 ATPs in my overall reaction. So pay attention that we are going through these steps twice per glucose molecule. And I will also lose a water molecule to form a double bond during these one of these 5 steps, which again, it's not necessary for you to understand which step that's going to happen. Be very careful not to confuse ATP production via substrate level phosphorylation with oxidative phosphorylation. So whatever happens during glycolysis and the way we make or regenerate ATP or ATP synthesis is via substrate level phosphorylation. It is very important for you to understand this concept because in another video, I'm going to discuss what's the difference between these two, oxidative phosphorylation and substrate level phosphorylation. But for the purpose of this video, I will define briefly what substrate level phosphorylation is. So basically, it is an enzyme catalyzed formation of ATP with direct transferring of one phosphate group to ADP from an intermediate substrate. So as I discussed earlier, if you look at the first step, I am grabbing one of the phosphate groups in ATP molecule, attaching it into my substrate, which in this case is glucose, to give me glucose 6-phosphate. This is not generating, I am using one of the phosphates in the ATP. But take a look at the 5 steps below, the 5 arrows that I've drawn. As I told you, I haven't drawn the intermediates and enzymes involved, but ATP synthesis during these 5 steps uses an enzyme. So that's why it is enzyme catalyzed formation of ATP, and we are directly transferring phosphate groups from our substrate, which are going to be those intermediates not shown, to my ADP molecule in order to form ATP. One of the common questions on the DAT is, where is the location of glycolysis? So, if this is my cell, and here's my cytosol, the glycolysis happens in my cytosol outside of my mitochondria. And this is the end of this video. I hope you enjoyed it. Have a wonderful day.